Greetings and welcome everyone to our sheep brain dissection. So today we're going to take a look at the sheep brain. We're going to talk about some major parts, some of the more important structures of the brain. We're also going to talk about some differences between sheep brain and human brain, and then we're going to cut it open and look at different angles of it. So we're just going to get started here by looking at some of the more important structures externally before we do cut it open. So the first thing I want to talk about are some of the differences between a sheep brain and a human brain. And I think one of the first and most obvious differences that you could probably tell based on looking at diagrams as we learn this in class is that sheep are obviously quadruped animals, right? They're on four legs. So their cerebellum and their spinal cord actually lie horizontal, right? Because their spinal cord is going to be going along their back. Whereas for us, ours would actually fall a little bit more like this, right? It would actually fall down. We'd have our cerebellum and stuff below rather than behind. So there's a couple other things I want you to see externally here, such as these sort of like these hills and valleys in the brain. That's super important. So let me grab my probe and we'll point out some of these external characteristics before we cut it open. So looking at the brain, the most obvious thing that you can see are the hemispheres, right? The cerebral hemispheres. Remember, the right hemisphere controls the left side of the body and the left hemisphere controls the right side of the body. And you can also see the cerebellum back here, which is there to control coordination, balance, and some muscle movement. And the spinal cord is just relaying information to the peripheral nervous system, right? It's sort of a go-between. But what I want to talk about are these hills and these valleys. So looking at the brain, you can see these things that kind of look like worms, right? Now, we did talk in class in lecture about three coverings of the brain. You can't see all three of them here. You can actually only see the thinnest innermost layer called the pia mater. The arachnoid layer and the dura mater were both removed when this brain was removed from the skull cap of the sheep from which it came. So the pia mater, that thin layering, is actually what's holding in these things that look like worms in the cerebral hemispheres. And those actually have a special name. So the ridges, the things that are coming up, like the folds, the worm-like structures, are actually called gyri, or a gyrus would be a single one of them, Gyrus. And the way that I always tell students to remember it is it actually comes from the same Greek word that gives us euro, or hero, however you pronounce it, Gyro, that you might have if you've ever been to Taste of the Windy City or another Greek restaurant. That little, that pita folded, wrapped sandwich that often has lamb meat and stuff in it. And the reason that's a really good way to remember it is that's what Y or GYR, that prefix, that etymologically that word means fold or roll from Greek. And that's actually what you're seeing here. You're seeing a fold or a roll. Now, you can actually see where there's all these blood vessels on the brain, all these blood vessels that are feeding the brain. There's probably nerves in there too, I could say, so they are also innervating. These blood vessels, though, lie in these valleys, these little cracks, and those are called sulci, S-U-L-C-I, and one of them is called a sulcus. So that's some external things that you can see. If we flip the brain over, though, there's a bunch of other features of the brain that I do want to show you on the back. This brain seems a little bit better than this one, so I'm going to actually focus a little bit more on this one. Right here on the underside of the brain, well, actually, let's talk about main regions. So from here down, you can actually see there's actually a little bit of a color difference. Anything below the cerebellum, so if you find the cerebellum and go to the bottom of the cerebellum, anything below the cerebellum is now spinal cord. But above that cerebellum, where you get to this lower portion of the brain right here, from here to here, right, all of these structures are actually part of the brainstem. And the brainstem is made up of three components. It's made up of the midbrain here, and then there are really two big humps. There's the pons and there's the medulla oblongata. So we've got midbrain, pons a little bit lower, and the lowest portion of the brainstem is the medulla oblongata, located right underneath the cerebellum. Now above that, we have a couple other important structures. We've got olfactory bulbs right up here. Those actually help us to smell. And then something you don't see on either of these brains, which is actually a bit of a disappointment, is the optic chiasm, this X that normally appears on the brain. So let me grab another brain that I have here. I apologize, by the way, if you hear a lot of background noise. It is Mother's Day, and my wife and my kids are playing upstairs, and so they are sort of a little bit wild. Now you can't see it so much on this one. This one does have a little bit of the dura mater attached, so I thought that that was kind of cool to show you. This one's actually already been mid-sagittally cut too, but I thought we'd be able to see the optic chiasm, and I'm kind of disappointed that you can't. If I can find a picture online of a really good intact optic chiasm, I'll try to throw that picture in right about now in the video, but it normally looks like this X, and it's crisscrossed because that's actually where the eyeballs attach, and each eye actually feeds your brain's opposite side to send it to the back here to the occipital lobe for processing. So I think those are all of the external structures that I did want to talk about. Just trying to think if there's anything else before I jump into it. I don't believe so. Just a bummer that we couldn't see the optic chiasm, but we could see the olfactory bulbs, which is really cool. Oh, another thing to remember about the olfactory bulbs that we did learn in class is that the olfactory bulbs, since they are directly connected to the frontal lobe, they are the only one of our 5 main senses that is not sent to the thalamus first. And I'm going to show you the thalamus. The olfactory bulbs sends smells directly to the frontal lobe for processing without having to go to the thalamus first. The rest of our senses, touch, seeing, hearing, and taste, all have to go to our thalamus first. So before I cut into the brain to actually show you the structures that we can see inside, I did want to show you one other thing. So I'm actually going to shave a bit of brain off here. And if you look really closely, what's really cool is you can actually see that the brain has multiple colors to it, right? You can actually see this really light color right here. That's called the white matter of the cerebrum. And then there's another shading right there called gray matter. And the reason we have white and gray matter actually is something I'm going to get into, and I'll probably try to shoot that video in here as well too. I'll try to draw that for you. Right here, we also have white and gray matter of the cerebellum. And you'll be able to see that too when I cut the brain in half, that there's light and dark sections of both of these of the cerebrum and the cerebellum. So I am now going to make a mid-sagittal cut. Since we've identified everything on the superior and inferior view of our brain, I want to look mid-sagittally now. So we're just going to separate the 2 hemispheres, and we're going to cut through something called the corpus callosum. The corpus callosum is nervous tissue that actually connects both of the hemispheres and allows both sides of your brain to communicate with one another. So we're going to cut right through that corpus callosum, try to make it as clean of a cut as possible. Remember, I do have another brain that I've already cut in half, so if for some reason this brain doesn't look amazing inside, we'll refer to that one. And as I open it up, there we go. So the part that I just cut through, the corpus callosum, which remember allows for communication between both hemispheres, is this part right here. That's the corpus callosum. So actually this does look pretty good opened up. It looks pretty symmetric. I think there probably still are a couple spots on the other one that might be a little bit more visible, but I think I can show you here. So structures when we're looking inside that I do want you to know about. So first of all, it's important to know, we talked about the midbrain before. The midbrain is still found right here. So this section right here is midbrain. And then right under it, you can see this little hump, that's the pons. And then pretty much the entirety of what's below the cerebellum is the medulla oblongata. So I've got midbrain here, I've got pons, medulla oblongata, and then remember anything behind the cerebellum and further down, that is the spinal cord. All right, I'm trying to think what else. Talk about some functions. So remember the pons is there for respiration. It controls arousal. It also is the go-between, remember, between the cerebellum and the cerebrum. Now the medulla oblongata, because it's so much further, this develops first. The cerebral hemispheres develop last. But the medulla oblongata is incredibly primitive, right? It's there to help with heart rate. It helps with defecation and vomiting and swallowing and all those really basic things that you do from the moment that you're born. I did mention the corpus callosum already, but what you can see inside the corpus callosum, where my fine-tipped probe is currently right now, is this cavity, right? There's actually a ventricle inside of the brain. And that ventricle is called the lateral ventricle. All right. It is possible, by the way, for that ventricle to become essentially poisoned by bacteria and stuff like that. You've probably heard of bacterial meningitis. So the meninges outside of the brain and the ventricle on the inside would actually become contaminated with a special type of bacteria to give you bacterial meningitis. Now, I'm going to point right toward the center, and I'm actually going to leave the probe in there. That spot right there is a spot that I've talked about already. And it's incredibly crucial to the proper functioning of the brain, because it is really the relay center for every, remember, your brain is really just a processing unit. So it's got to get messages from your sense organs, your eyes, your nose, blah, 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 blah. Well, remember, your nose is an exception, but all the other sense organs send things to this structure first, and that's called the thalamus. So the thalamus is your relay center. Once those messages are sent here, right, if it's coming from your eyes, it's going to go back to the occipital lobe. If it's coming from your skin, it's going to go to the parietal lobe. If it's coming from your ears, it's going to go to the temporal lobe. So depending on what sense organ is sending the message, it will always go to the thalamus first, with the exception of smell, remember, and then the thalamus will sort it out, kind of like USPS. Now, again, I'm a little bummed. I'm going to see if I can show you on another brain piece over here. Oh, yes, I can. Okay. So on the one that I was just holding up, there's no little bump on the bottom because we didn't have it intact, but this one looks great. This thalamus, by the way, looks really great on this one too, and you can clearly see the white and gray matter of the cerebellum on this one. I think the one that I just dissected, you could easily see that too, but this one has just a little bit more definition. And the definition, by the way, in the pons is just extraordinary. Look at that pons on this one. This piece right here, though, is the piece I wanted to talk about next, and that's called the optic chiasm. This little bump is actually where the optic nerve would attach, and you'd have the eyeball sending messages. Again, once it comes in on the optic chiasm through the optic nerve, it's going to go to the thalamus first, and the thalamus is then going to push it back to the occipital lobe for processing. So there's only a couple other structures that I want you to know. Remember, if this is the thalamus here, then the hypothalamus is right below it, because hypo means below. And then one other structure, now on this one, it looks monstrous, but I'm not talking about that structure. So I'm gonna see if I could point it back out on this brain. Here we go. So if I go back to our brain that I was looking at just a moment ago, and I find the thalamus again, right in the middle, right behind the thalamus, there's this little teeny structure right here. Look at how small it is. You'll notice it does kind of look like a little pine cone, and that's what gives it its name. That right there is the pineal gland. And the pineal gland, remember, the purpose of the pineal gland is to secrete melatonin, which helps with our circadian rhythm and our sleep cycles. So I'm trying to think. I mean, there are other structures of the brain. I could talk about all the lobes, the frontal lobe, the parietal lobe up here, the occipital lobe, and then you can't really see it here, but you can see it here. That would be our temporal lobe. But I think those are all the main structures that I want you to know as we dissect our brain. And yeah, I think that is about it. So thank you so much for your time. I hope you enjoyed this walkthrough of our sheep brain, and I hope you learned something. Thanks so much. Take care.